So first of all, CytoGPS is a tool that a postdoc of mine developed during his PhD thesis, which takes karyotypes, which are written in a standard text-based notation, and converts them into binary vectors that are easier to analyze with a computer. And part of the motivation for this is there are many databases, especially in leukemias, CLL in particular, of what the karyotypes of patients are. Probably tens of thousands of them in some places, and this is data that is being ignored. It's used clinically immediately, but nobody's going back and doing research on it. So CytoGPS, by turning it into binary vectors, lets us apply all of the sort of modern machine learning technologies to karyotypes. And so we can identify groups of samples who have similar patterns of abnormalities. We can find which abnormalities are driving different cases. And so what we've done is we've added some of the standard unsupervised learning technologies to CytoGPS so that we can do what are the clusters and what are the abnormalities that drive them. And again, what this does is it lets us not only see the very common ones, the trisomy 12s and the deletion 17Ps and the deletion 13Qs, but we can burrow deeper and find rare abnormalities there. The only way anybody's ever found recurrent abnormalities in CLL is by visual inspection of many karyotypes, which is tedious and error-prone and hard. What we've managed to do by applying CytoGPS to the set of all CLL karyotypes in the Middleman public database, that's everything curated from the literature since 1970, is that when we burrowed deeper, we found an abnormality on 12q13 that is rare but still consistent. It shows up as a fusion in our data, but fusions include small deletions or small amplifications. And so what's interesting about this, of course, is that when you're trying to figure out which genes are driving trisomy 12, you have to look all across chromosome 12. Now at least we have an idea that we should look in 12q13. That gets us down to about 10% of the 1,000 genes we would have had to look at. So we think that that's useful because it will potentially give us more insight into what that particular form of CLL comes from and which genes are driving. Now, at this point, CytoGPS doesn't have direct clinical applications. It's more a research tool to understand the biology of the disease. The gene expression signatures, on the other hand, we think potentially have clinical applications, in part because we still think that chemoimmunotherapy is good for some people. If we can identify patients who are going to be cured or have 15 years in remission with FCR, which is a small number of cycles of therapy instead of continuing treatment with some of the modern ones, that could be of benefit.